Chapter 6, Section 12, Protein Secretion, the Sec and Tat systems. Okay, pretty much all the proteins are going to be made in the cytoplasm, okay, especially we're talking bacteria, okay, it's the only place they can be made, cytoplasm. Well, many of these proteins, or a lot of these proteins, need to be outside the cell. How do they get out? If you're looking at toxins or exoenzymes, or even the enzymes that are there for like autolysin for, cutting up the cell wall so you can go through and do some remodeling of the cell walls and things like that. Well, if it's a protein, if it's some sort of organic compound or whatever, it's going to be made in the cytoplasm and then it has to get out. Well, big things going to need big transporting systems. Big transporting systems are going to need energy, ATP, GTP, or even proton motive force. You can start with translocases. These are big transports, proteins that are big in bacteria and archaea. They take unfolded proteins, insert them in the membrane, and then push them through. Once they're through the membrane, they will, finish their folding. TAT system takes folded proteins and then passes them through the membrane. Sec for secretory, TAT, which I forget what that stands for. How do the cells know which ones stay in, which ones move out? Well, there's going to be an N-terminus signal. N-terminus, this means amino acid number 1 has the amino group sticking out. You'll see the C-terminus, that means the last amino acid has the open carboxyl. N-terminus, C-terminus. So at the N-terminus, there's going to be about 15 to 20 residue sequenced there. That is a quote-unquote signal to enzymes, transport proteins within the cytoplasm of the bacterial cell. There's going to be versions of this in us eukaryotes too, that will tell certain proteins to come bind here. If those proteins combine, well, they're going to target that polypeptide with this specific signal to different places in the cell or to different transport proteins in the cytoplasmic membrane. The signal sequence, as the things are being moving, and also inherently there's going to be certain signals, certain sequences in there that are going to keep this polypeptide from its final folding, making the toxin in the cytoplasm. It's going to wait till it's transported out, and certain parts of it are clipped in the process until it gets to the final folding outside the cell. We'll see in many things, export may begin before the protein's finished. So the messenger RNA has the ribosomes marching down it, and the ribosomes may be in close proximity to the cytoplasm. So as the polypeptide's being made, the first 15 to 20 amino acids are being made, proteins are going to recognize it as a signal sequence for excretion outside the cell using the Sec transports or the TAT. It's going to target it, and the ribosome is going to be right there near the cytoplasmic membrane as the polypeptide's coming out. It's being pushed through the cytoplasmic membrane and already out the door, as it's showing right here. Signal peptide, signal recognition particle is going to target it here, and it's going to immediately start pushing it out using Sec-A. Sec-A here, going to target it, push it out, and when it hits the periplasm, here's what it'll be cut, pasted, twist, tweaked, whatever it needs to be, whatever it needs to do to itself to get to the final secondary, tertiary, quaternary structure, and you now have a working enzyme. If it doesn't have a signal sequence, well, it's just going to stay in the cytoplasm. None of this matters. So you have Sec-A, you have some other signal recognition particle that's going to drag them out. Drag them to the cytoplasmic membrane, and then somehow push them across to get them out. So Sec-A in the cytoplasm, I don't know why that has periplasm there. Sec-A is in the cytoplasm. Or signal recognition particle are going to grab, and they're going to transport it to the cytoplasmic membrane, where there's going to be some form of translocase that's going to push them out. If it's going to be the signal recognition particle, Sec-A takes a completely formed polypeptide that hasn't folded yet. The SRP particle, well, this is going to chaperone that growing polypeptide to the cytoplasmic membrane, but it's bringing along the ribosome along with it that's still marching down the messenger RNA. The TAT translocase, twin arginine translocases, this translate cofactor containing. These are going to be things that are like iron sulfur, cytochromes, respiratory enzymes. So these are going to be different types of proteins that are going out into the environment and are picking up stuff. Iron, are picking up, molecular iron, are picking up molecular sulfur, things like that, to be brought back to the cell. Or they're going to be some form of a protein that's going out and going to start modifying the microenvironment. The TAT-BC proteins recognize the signal sequence. That the TAT-A, which is the transmembrane transporter. So TAT-BC binds the polypeptide, brings it to TAT-A, TAT-A, then transports over these cytochromes and whatnot. You look at these are going to be big, bulky things.